Okay, so no question, so I will proceed it. Okay, so normally synapse, I think everyone is already know, right, what is the synapse that you already learn in SPM2. Okay, anyway, okay, if you already forget, never mind, you make a revision again lah. Okay, so normally synapse is the junction or the gap between the two neurons, like the sensory neuron between the sensory neuron and interneuron between and the motor neuron. So all these is a gap we known as synapse. Okay, so you see that this is one A neuron and then this is opposite neuron, B neuron. So this is what we call as presynaptic axon and postsynaptic cell. Presynaptic neuron and also postsynaptic neuron lah. So presynaptic is the electrical impulse will be coming in, and then postsynaptic is receive the electrical impulse from the presynaptic axon 1. Okay, so normally you need to know the structure. So neuron that terminate synapse is presynaptic, and then terminate is presynaptic, and then begin at synapse is postsynaptic, and then shrilling end is known as synaptic knob or synaptic cleft, is both are acceptable, and then separator or synaptic knob containing synaptic knob is at this one is we call as synaptic knob, sorry, and then the gap between the neuron is we call as synaptic cleft, and then cytoplasm containing mitochondria lah, where we know that because it require energy, and then we know that it containing neurotransmitter, right? Neurotransmitter, because cytoplasm containing neurotransmitter. So normally in the presynaptic neuron, synaptic membrane, synaptic knob, the neurotransmitter is enclosed in the synaptic vesicle. Okay, so this is just a general structure about the synaptic structure lah, synapse lah. So this one I think I don't need to wasting the time at here. So impulse transmission, I think this one is you are also learn in SPM2. So normally if you talk about the impulse transmission, so normally when the electrical impulse is already reached the synaptic knob, when it already reached the synaptic knob, so that it will trigger the synaptic vesicle to release the neurotransmitter. But before that, you know that depolarization and repolarization process, right? So action potential still will be occur. So when the action potential is occur, done. So depolarization will cause the calcium ion channel inside on the presynaptic membrane open, and then calcium ion will be influx move into the cytoplasm. Yeah. So when the calcium ion is already inside the synaptic knob, so that the calcium ion will cause the synaptic vesicle fuse to the presynaptic membrane and starting to release the neurotransmitter. So any type of neurotransmitter, so just now I already mentioned, right? One of the example is acetylcholine and another one is noradrenaline, right? Okay. So this neurotransmitter later it will bind toward the receptor that located on the postsynaptic membrane. Yeah, so you see this diagram. So it will bind to the receptor at the postsynaptic membrane, and then it will trigger the action. So not necessarily about the sodium potassium pump, not necessarily. It can be have another type of reaction. But one of the example is trigger sodium potassium pump lah, meaning that sodium ion is pumped into the cell and then potassium ion is pumped out from the cell. Yeah, to trigger the action potential of the postsynaptic membrane. Why? Because postsynaptic membrane. Okay. Okay, so the another one, yeah. Okay. And then this is the explanation, so I don't need to explain much. Okay. Okay, so I need to explain about the activated receptor with same impulse to postsynaptic neuron. Okay, So when the neurotransmitter is already bind to the receptor on the postsynaptic membrane, it will trigger the action potential of the postsynaptic neuron. So this receptor will send the impulse to the postsynaptic neuron to trigger the action potential, and then the impulse that we will have is two types of impulse that we need to learn is Epsp and also Ipsp. Epsp is excitatory postsynaptic potential, and also Ipsp inhibitory postsynaptic potential. So excitatory Epsp, you will see the excite, so meaning that it will change the membrane potential change, will trigger, bring the closer to firing, meaning that this one will trigger the action potential one of the postsynaptic neuron. But for this one, it will inhibit the action potential for the postsynaptic neuron, meaning you see that action take the neuron farther away from the firing level. So firing level actually is we call it action potential lah. Okay. So in Epsp, so normally the neurotransmitter role is acetylcholine. So normally it will bind to the receptor and permeable to both potassium and sodium, and then the sodium ion will be influx into the membrane to depolarize it, so that it will trigger the action potential. But Ipsp is different. It would inhibit the action potential, meaning that you see this. One of the example of the neurotransmitter is GABA. Later we will see it, the full name GABA. So this neurotransmitter is reversible compared to acetylcholine, because acetylcholine is trigger action potential, but GABA is inhibit action potential, meaning that it will more permeable to the ion we call as potassium ion and also chloride ion. Okay, when this ion channel are open, the postsynaptic membrane is hyperpolarized. Oh, you will still remember about the hyperpolarized, meaning that it will more negative compared to resting state. So it will totally inhibit the action potential of the postsynaptic neuron. So it will move the membrane potential further from the threshold level, and then inside the cell is negatively charged, so no action potential will be triggered. This is what we call as Ipsp. Okay. And then after the neurotransmitter is released out from the presynaptic neuron, then the neurotransmitter cannot always maintaining at the in the synaptic cleft. The neurotransmitter need to recycle back. Dia kena hilang punya. Dah tak boleh selalu dekat. Cannot always maintain at the synaptic cleft one. It need to recycle back or disappear one. If the neurotransmitter is still maintaining at here, meaning it will trigger the action again and again. Yeah, because neurotransmitter is still maintained at here, so it will bind again. You need to know that when the neurotransmitter bind to the receptor on the postsynaptic neuron, once the action potential is triggered, this neurotransmitter will be loose by link. Dia akan keluar, but dia kena disappear daripada synaptic cleft ni, hilang daripada synaptic cleft. If not, it will trigger again and again. So you need to terminate the neurotransmitter signaling. So there are two ways to terminate it. Either using enzyme, you use the enzyme to break down it, or the neurotransmitter automatically flow back into the presynaptic neuron through the transport channel. Two ways: either using enzyme or using the neurotransmitter transport channel, recycle back to the presynaptic neuron. Two ways lah. Okay. And then about the summation or postsynaptic potential. So this is about the Epsp and Ipsp that learned just now. Okay, so normally in synapse, in the postsynaptic neuron, if you want to trigger the action potential, you still need to make the action membrane potential reach the threshold level. Then you can trigger the action potential in the postsynaptic membrane. But normally in the synapse, due to the synapse got the gap, right? So normally the impulse at the postsynaptic membrane is quite low compared to presynaptic neuron, because it already got the gap there. So the electrical impulse normally is very low. It is very low. So even though when your electrical impulse is Epsp2, like this Epsp, but if your electrical impulse is very low, that not reach the threshold level, then postsynaptic membrane also cannot trigger the action potential. Also cannot trigger the action potential. Only when your membrane action potential reach the threshold level, then it will trigger the action potential on the postsynaptic neuron. So how to trigger it? You will see this first situation, no summation, meaning that the impulse from E1 and E2, you see E1 and E2 is different, is only from E1 only, meaning that the nerve impulse is only coming from first part only, this part only. Other type of dendrit does not receive any impulse, only E1 receive impulse, and then the impulse received is separate time. The first impulse is maybe at 1 second, and another 5 second E1 still receive the another E1 impulse, but these two impulse is too far away. The period for this both impulse are too far. Like maybe the first E1 come, but another E1 need to wait about 30 seconds. So when you too long, the duration, the gap is too long, so it cannot reach the threshold level, so it cannot trigger action potential for postsynaptic neuron. You need to know this action potential is triggered for postsynaptic neuron. Okay, but if they are the temporal summation, meaning that when there are two types of the nerve impulse coming from the same synapse, same synapse, and then the impulse, the gap between these two impulse are very short, so maybe 2 seconds, 1 second, or 0.5 seconds, very fast, so that it can combine it together and it will reach the threshold level and trigger action potential. So this one is we call as temporal summation. This one will only involve one type of synapse only, one synapse only. But there are also the spatial summation, meaning it involves two synapses. So maybe one synapse at here and one synapse at here, and they combine together at the same time. You need to measure at the same time. If two get like this, then no also lah, no action potential. But if at the same time too, we know as a spatial summation, so that it will reach the threshold level and trigger the action potential of the postsynaptic neuron. Final one is partial summation, but it will involve Ipsp. So when it involves Ipsp, you know that Ipsp is inhibit the action potential. So once it be involved, although you have Epsp here, but since got Ipsp at another part, so it will inhibit the trigger coming out. Coming out is depolarization, right? Depolarization, right? Why? Because why? Because in depolarization process, sodium ions coming in, right? And coming in, right? And then you need to maintaining the high positively charge inside the cell. So potassium ion channel, another way is potassium ion channel can be open and coming out, so that it can increase the rate of depolarization and increase the permeability of potassium ion to the membrane. And another one is noradrenaline. So normally this one function is to involve in parasympathetic nerve effect. So normally this one is involve the CMP activation, and then it will get the calcium ion to stimulate it, and then it will trigger the depolarization process. And another one is dopamine. So dopamine actually is not in our syllabus, but later you will learn about the cocaine mechanism. So you should know too lah. So it is one type of neurotransmitter that normally is play the role in excitation and inhibition process. So normally this one or neurotransmitter can be inhibited by cocaine. One type of drug, right? Cocaine, I think everyone is familiar with it. What is cocaine? So later we will learn about the cocaine drug mechanism inside in our nervous system. So dopamine is one type of the neurotransmitter that can be inhibited by cocaine. Okay. And another one is GABA. This one I just want to show you about the example of Ipsp only, inhibitory summation process that I mentioned before. So the full name is gamma aminobutyric acid, GABA. So maybe in essay question, if you essay question or structure question ask you about the example of the Ipsp, you can write this GABA. Full name is gamma aminobutyric acid. So it is a inhibitory neurotransmitter. So it inhibit the reaction. So actually this is a mechanism, but actually you don't need to know it because it actually not our syllabus. But however, because it involves Ipsp, so you should know it